Welcome to today's lab video, where we will learn about properly conducting a dilution experiment. A dilution is the process of reducing the concentration of a specific solution by adding in more solvent, which in most cases is water.
In this photo, you can see the end results of today's lab. On the left, you have a properly created dilution, and on the right, you can see the original stock solution. In case you were wondering, a stock solution is a previously prepared liquid that has a known concentration, and a dilution is the less concentrated liquid we will create from the stock solution in today's lab.
In order to make the dilution, we will need to use the dilution equation. This equation consists of four variables, which are as follows: initial concentration, initial volume, final concentration, and final volume. To solve the equation and complete our experiment, we will need to start by taking a sample of the stock solution. Note the original sample's volume and stock concentration, as these will become our initial volume and initial concentration measurements. Afterwards, add a specified amount of water to your original sample and record the new volume, which becomes our final volume measurement. Now that we have three of the equation's four variables, we can algebraically solve for the unknown variable. In this case, it's the final concentration.
But before we can begin our experiment, we need to ensure that every lab member is wearing appropriate lab attire. Make sure everybody has on long pants, closed-toed shoes, and a shirt that properly covers their upper body. And don't forget to always wear your safety goggles.
Begin the lab by transferring some of the provided stock solution into a smaller container so your group can use it throughout the experiment. Make sure to note the stock solution's predetermined concentration, as you'll need this for the initial concentration variable in your calculations later.
Start by using a pipette to move over a certain amount of the stock solution into a volumetric flask. Using the meniscus, make sure to note the volume you originally drew up. In this case, the volume is around 3.93 milliliters.
Next, transfer some of your solution into a volumetric flask and observe the pipette's new meniscus to calculate the pipette's final volume. In this video, we can see that pipette's new meniscus sits at 7.0 milliliters. To determine the amount of solution you deposited in the flask, subtract the pipette's final volume from its initial volume. This number will be later used as the initial volume of the stock solution.
For those that aren't familiar with the concept of measuring volumes with a meniscus, here's a brief explanation. A meniscus is a small bowl-shaped dimple that appears on the surface of liquids when they are surrounded by a container. This phenomenon occurs because of the forces of adhesion and cohesion battling one another. In the case of a meniscus, the adhesion force wins out as the liquid's molecules are more strongly attracted to the walls of the container than they are each other. Because of the meniscus's concave shape, the liquid you are measuring may seem to be at different volumetric values depending on where in the curve you look. To solve this issue and get an accurate measurement, always make sure to measure a volume sample at eye level and to take the actual reading from the bottom of the meniscus. In this example, you can see that the bottom of the meniscus sits at 4.70 milliliters.
Next, we will begin diluting our stock solution. This is done by adding water into the graduated flask that holds our stock solution until the flask's marked volumetric graduation is matched. This allows us to confirm the total amount of volume inside the container, which we will use in our dilution equation as the final volume variable.
As you can see, the meniscus of our dilution matches with the flask's graduation mark, thereby confirming that our final volume is equal to 25.0 milliliters.
Now that we have successfully created a dilution, we need to calculate its concentration using the dilution equation and the values we noted in our lab book throughout the experiment.
To use the dilution equation, input the data you collected during the experiment into the proper variable slots and begin by simplifying the left side of the equation. Afterwards, divide the simplified left side by V2 and you have your answer. In the case of sample A, the dilution's concentration was 5.28%.
With this new dilution data in mind, many other scientific concepts can be explored, such as the relationship between density and concentration seen here.
Before you pack up and leave, there is one last thing you need to do. You must clean up your lab space and dispose of the materials you used.
For this experiment, you can simply wipe down your lab bench with a damp paper towel. After that, make sure to properly dispose of any excess reagents or solutions. For the substances used in today's dilution lab, it's okay to pour them down the sink.
Thanks for watching, and we hope you had an amazing lab experience.